Hello students, how are you people? Hope you are all doing good. Okay, in the last class we are discussing about mainly the structure of eye. Yes, in the last class we studied that the details of the eyeball. So as such, the eyeball has the two important chambers. What they are? Yes, it has mainly the outer, we call it as aqueous chamber, the inner, behind the lens it is vitreous chamber, we have discussed it. The wall of the eyeball, yes, it is made up of three layers. Innermost is the retina, middle is mainly what we call as a choroid, outer is the sclera. Modifications of the retina, they have the rods and cones which are mainly photosensitive. And then choroid modification, yes, it is mainly ciliary muscles also as well as iris. And then outermost sclera, yes, we call it as the white part of the eye, that is the sclera. So these things, yes, we have discussed. In today's class, we will study about mainly some more structural details of the eye and the functioning of the eye, how the image is created in the eye we will discuss, as well as we will study about the certain disorders of the eye. So now, looking into the human eye, yes, you know very well, human eye, it is very attractive, having the structure like this. Yes, it is well accommodating to the different intensity of the light also. Yes, the human eye is a spindle shape and well protected by the eyelids as such. And here inside we find it is mainly, yes, the lens surrounded by the iris muscles, and this is called as mainly the pupil. It is the opening through which the light enters inside, and we find, yes, there are what we call as these are the circular muscles, as well as we find there are number of longitudinal muscles. Okay, these circular and longitudinal muscles, they are mainly the iris muscles as such. So what we find here, these are all the iris muscles. These are also what is iris. And then this is the white part, sclera, and then the corner or the edge of the eye, it is called as yes, conjunctiva. Redness of this part is also called as conjunctivitis. And then when we are very much tired of, and then there is a certain kind of what you can do secretions that become the hard, okay, dust like particle, yes, they accumulate, and that is called as a crust. So in the early morning, yes, you will find the crust because the eye, mainly lacrimal glands, secrete that to eliminate or remove out whatever we can say, mainly the waste particles. So here we find mainly this is the eyelid, and then the eye is well protected by most attractive long hairy structures, and what we call them as mainly eyelashes. Okay, so camels have more long eyelashes than a human. So these eyelashes are, yes, very important, that keep preventing the entry of the dust particles. These are the hairy structures, we call them as eyelashes. So we need to understand how actually the light gets accommodated. So when we talk about the eye functions very well, mainly to the high intensity light, high intensity light, as well as low intensity light as such. So in both the cases, mainly the structure of the pupil, yes, here we find in the high intensity light, mainly the pupil become very small and narrow pupil. So yes, how it is possible? It is going to become narrow pupil, and then here what we find mainly there are the circular muscles. So circular muscles contracting and longitudinal muscles are relaxing. This is the condition during the high intensity light. So you need to remember circular muscles of iris contracts and longitudinal muscles relaxes. Longitudinal muscles relaxes or relaxed. So this is the condition that can bring narrow. Yes, the pupil become narrow and it accommodates very less amount of light, which is that much only required during high intensity light. Our eye or the lens doesn't allow too much of light entry. It is mainly the amount of light. Yes, it is controlled by the iris muscles. So iris act as an aperture and help in the accommodation of less amount of the light when there is a light intensity is high. Now, when light intensity is very low, so now here what happens to the pupil? Let us understand. So the pupil become widened. So we can say that wide pupil. Why it is mainly wide? Because of let me tell you, relaxation of the circular muscles. So circular muscles get relaxed. Circular muscles relaxed. Whereas longitudinal muscles are contracted. So longitudinal muscles contracted. Longitudinal muscles contracted. As a result, what happens? The pupil is wide and allow more amount of light that can enter. This is called as light accommodation in the eye. When the light is more or intensity is very high, the eye doesn't require that much of too amount of light to enter into the eyeball. So there, what will happen mainly? There is a relaxation of longitudinal muscles, mainly contraction of circular muscles, creating narrow opening for the pupil, so less amount of light will be entering. And then suddenly from the bright light you are going inside the dark room. So then what will happen? So here, yes, you are observing mainly when you enter into the dark room, your eye needs to take out more, so to allow the more light inside. So the widening of the pupil, how it is resulted? Mainly by the relaxation of the circular muscles, contraction, pulling off mainly the circular muscles to the periphery done by mainly contraction of longitudinal muscle. So that widening of the pupil, more amount of light accommodated inside the eye. Then it takes some time to see the things very clearly when you shift from the bright outside light to the dark room inside. So this is called as light accommodation, mainly yes by the iris muscles, and the iris muscles act like a aperture. Now we need to know, yes, our eye discriminate red, green, blue color. So in the nature, whatever the color we see, everything is mainly made up of a combination of red, green and blue only. So RGB is called as a basic color. Now this color discrimination, how it is done by our eyes? Why? Because our eye has a most important photosensitive layer called as a retina. So in the retina, yes, what we find? Rods and cones. Let me tell you, yes, the photosupigmented, yes, photopigmented sensitive, yes, the sensitive layer, we call it as retina. So this retina is made up of two important cell types. What we call them as mainly the rods as well as cones. Now here rods, yes, the purplish color pigment they have, purple pigment. What is that called as rhodopsin? Rhodopsin. Yes, cones are having mainly a normal pigment called as iodopsin. This is the first difference. Then rods are somewhere 120 millions in number. Cones are 60 millions in number. Okay, yes. So they are sensitive to what we say mainly the scotopic light or dim light, whether it is early morning or late evening. Yes, mainly the rods are very active. These are sensitive to photic light, sensitive to photic light or daylight or bright light, okay. And yes, when we talk about mainly the rods cannot be able to discriminate the colors, okay. So we can say that can't discriminate colors, where the cones discriminate, they discriminate mainly red, green and blue color. So these are few basic differences between rods and cones. So as such, rods are cylindrical rod shape, cones are mainly yes cone shape. But cones are very very essential for color vision, whereas rods are essential for whether bright or dull images. Okay. So these are yes what we are observing mainly basic rods and cones. But if we go into the detail of mainly structure of retina, let me tell you, yes, I hope you understood the basic appearance of the human eye. This is what we say the structure, mainly iris muscles and lens is present. Around that white part is called sclera, the edges are called mainly conjunctiva. Yes, when there is a dust particle, anything dirt or unwanted substances, yes, the mucus accumulates there and then becomes solidified. That is called the crust of the eye, that will be detached or removed every morning. And then how the eye is adjusted itself to the bright light and low light because of the narrowness of the pupil caused by the mainly contraction of circular muscle, relaxation of longitudinal muscle. So in the bright light, less amount of light is allowed to enter in the lens. When there is a low intensity light, you are in the dark room, you want to see all the things clearly. So what will happen? Widening of the pupil is needed due to the contraction of the longitudinal muscle, relaxation of the circular muscles. And then the differences between the rods and cones, which are most important and essential cell types as such, mainly in the retina. So rods, purple pigmented rhodopsin it has, cones are mainly iodopsin. Yes, mainly rods 120 million, cones are 60 million. Rods are sensitive to scotopic light or dim light. Yes, cones are sensitive to the photic light or bright light. Cones cannot distinguish between the colors, whereas the rods distinguish between the colors such as red, green as well as the blue. Now let us understand basically. So mainly the structure of retina as such. So it will be very easy for us to discuss mainly mechanism of yes how the image is formed. So when we look into the structure of as such the retina. So retina structure reveals that mainly there are what we say these are the yes rod and then here is the cone. Again the rod, again the cone. Yes. So like this, very alternatively, because as such, this is the lens and here is the eyeball, and we see that here is mainly the blind spot and then the fovea centralis and then this. What we are observing, it is the retina, photosensitive layer. The light, yes, it will mainly come to this. What we can say mainly the layer. So here only the light reaches. So there what we say this is the retina. So mainly the retina in detail what we can see these are the structures in the retina. So there yes we can find these are the kind of structures, and then there is what we find mainly. These are called as bipolar neurons. Okay, so here we are finding mainly the rods, and here we are finding the cones, and we are finding here bipolar neurons. Okay, yes. Then they aggregate here, and this structure is called as a ganglion. And then further, yes, like this many ganglia and more number of nerves reaches, and they form common optic nerve. Optic nerve, this will go to the, yes, visual cortex of the brain, to the visual cortex of brain. It reaches further. So this is what we say the structure of retina means. Mainly it is called as the external, means it is now exposing to the light, that's why it is called external. Then this part is mainly the middle, bipolar neurons in the middle. Then there is what we can say mainly the optic nerves in the inner. So from outside to inside, what are the cell layers in the retina? Yes, more light exposing part is called the external. So it is rods and cones. Then middle is mainly bipolar neurons, inner side ganglions and optic nerve you find. So this is what we say the retina structure, which is very essential because the rods and cones receiving the signals of the light, sending that signals mainly to the bipolar neurons, then ganglion, then to the optic nerve, then it reaches to the visual cortex of the brain. Okay, so this is, yes, we need to understand how actually the basic structure of the retina. So let us understand now how this retina functions and mainly convert the light signal further into what we call it as mainly it converts the light signal into, yes, the impulses. So how those impulses are mainly received by what we say mainly the brain. So here let me mention as such, when the light is mainly falling on the retina, it is reaching the retina. So retina has mainly rods and cones. So the light is reaching mainly retina has rods and cones. They have what we can say that mainly there is a opsin. Opsin is a protein, and retinol. Retinol, yes, is mainly what we can say that it is aldehyde of vitamin A. So they get dissociated, yes, dissociates when exposed to light. So these two components get dissociated when exposed to the light. Further, what will happen? This dissociation changes the permeability of bipolar, yes, neurons and nerve fibers. There what will happen? This light signal now it is getting converted into chemical signal. So permeability changes means, yes, action potential generated, formed. This action potential further travels now. Yes. So the impulse transmitted further. Yes. To ganglia. To the ganglia then to the optic nerves, then to the visual cortex of the brain. So now what will happen? Brain will analyze this image signals. So now brain identifies the object by previous memory. This is very simple mechanism of what we say mainly the image formation and detection. First you need to remember the retina structure. So retina is basically having rods and cones, what you are observing. So these rods and cones, yes, mainly photosensitive. Rods are sensitive to the mainly dim light, cones are sensitive to the daylight and discriminate the color. So then the signals passing through the bipolar neurons, then to the ganglion, then to the optic nerve. So actual thing, when light is fall on the rods and cones, there is opsin, a protein, and then retinol is the aldehyde of vitamin A. Before they were associated, but they get dissociated in these cells. That lead to the change in the permeability of these nerve fibers and neurons. So action potential generated here that pass into ganglion, then pass to the optic nerve, then to the brain. Then brain identify what is the object we are observing. This is what we talk all about mainly the mechanism of formation of the image as well as the detection of the what we can say the things by the brain. Yes, mainly our eye converts that light signal into impulses and brain understands that impulse language. Okay, so this is the overall mechanism of what we say light falling on the retina, rods and cones receiving that their opsin and retinal associated, but due to exposure of light they get dissociated, and then this dissociation brings about change in permeability in the bipolar nerves, and then impulse generated, that is action potential that travels through the ganglion, then to the optic nerve, then reaching to the visual cortex of the brain. Brain identify what the thing we are observing based on the experience or previous memory. This is called as a mechanism of image formation or mechanism of vision, we call it as. Okay, so well, there are a few important things yes we need to discuss about the eye as such, namely how this normal vision works as well as what is mainly the certain kind of disorders related with the eye. Yes, we will discuss that also. So here when we talk about normal vision, we say as such, it is also known as emetropia. Emetropia means a normal functioning of the eye. So let us understand that. So in the emetropia as such, let me mention here normal image formation, where the image is formed. In fact, it is on the retina and it is called as mainly yes emetropia. Okay, normal image formation is also known as normal vision. Normal vision. Now here, let me tell you, in the emetropia, yes, we do have the lens, and this is the eyeball. Yes, so mainly the light rays are passing and creating an image on the retina. So image on retina is very normal thing. So this is what we say emetropia or the normal. Now there are certain yes the eye related disorders as such. Number one, yes, let me mention disorders of the eye. Number one, it is called as mainly myopia. Myopia is also known as the short-sightedness. Short-sightedness person unable to see the things of far objects. Okay, so he can see the things nearby, but unable to see the far objects. Unable to see far objects or distant objects. Okay, so what is the problem in this condition? Let me tell you. Yes, the person is having the lens and the eyeball is elliptical. Okay, so what is happening here? The image not formed on the mainly retina. Image is forming front of the retina. So yes, image front of retina, not on the retina. So because of elliptical eyeball. Now how this is recorrected? Yes, it can be recorrected by using the concave lens. So concave lens is used. Yes, and then the eyeball, no doubt it may be the elliptical, but concave lens make the image that has to form on the retina only. Yes. So concave lens and then yes, image on the retina. Image on retina means this myopia is recorrected by using the concave lens. This you need to remember. Concave lens to recorrect myopia. Okay. The second kind of disorder, yes, you need to know. What is that? Let me mention here the second type of disorder. Yes, it is also known as the hyperopia or hypermetropia. So here hyperopia or hypermetropia. It is, yes, also known as mainly farsightedness. So it is earlier it was short, it is a farsightedness. Person able to see the object of the long distance but unable to see the objects of near distance. So yes, unable to see near objects. Why? Because now here what is happening mainly, so the eyeball is very narrow. So let me mention this is the lens and narrow eyeball. As a result, the image is formed behind the retina. Okay, so this is the narrow eyeball. Narrow eyeball, and then image behind it. Image beyond, we can say, beyond the retina is formed. How this can be recorrected? Yes, by using the convex lens. So now, yes, what they do, they take the convex lens, yes, in front of the eyeball or the spectre. We say, yes, this eyeball is no doubt definitely maybe the narrow one, but when the ray passing through convex lens and the biological lens, the image formed on the retina. Image on retina, it is formed by using the, yes, convex lens to recorrect what we say hyperopia. This is mainly the kind of what we say the disorder. Yes, one is myopia or short-sightedness, unable to see the far object but able to see the nearby, and the eyeball is very much elliptical, image is forming in front of the retina. So to make the image to fall on the retina, concave lens is used, whereas there is a very narrow eyeball, okay, developmental defect we can say, and then the image is formed beyond the retina, that is called as hyperopia or hypermetropia or farsightedness. So unable to see the nearby object. So this can be recorrected by using the convex lens, where it makes the image to be formed on the retina itself. Okay. So this is what we talk about, mainly hyperopia. Then there is one more disorder called as number 3. We say glaucoma. Glaucoma is mainly excess aqueous fluid. Where is the aqueous fluid located? In the aqueous chamber, means in front of, yes, here aqueous fluid is present. That is excess, and then excess aqueous fluid leaks, yes, into vitreous chamber, and this causes glaucoma. So as a result, ocular pressure in the eyeball increases. So yes, mainly ocular pressure increased, and the condition is called as the glaucoma. Okay, there is one more disorder, astigmatism. Astigmatism. What is this astigmatism? Means mainly here is the lens, but the cornea and lens are dislocated. This may be normal. Imagine this is a normal condition, but here is mainly the cornea, but the lens is disoriented. So this disoriented, we can say disoriented lens and cornea, that causes blurred image. Okay, astigmatism leads to blurred image. Okay, due to the, yes, disorientation of the cornea and lens. This is called as astigmatism. Well, there are some more, yes, other disorders. Let me mention that also. Here, when we look into other disorder, I hope you understood, mainly regarding the emetropia, myopia, hypermetropia, you understood. Let me tell you the remaining disorder type. Yes, there is one more called as number 5th one, presbyopia. Presbyopia means, yes, lens loses its elasticity. Means the lens is capable of contracting and relaxing because it's made up of crystalline protein. That loses its elasticity. As a result, a person will have a blurred image formation. So he cannot see the things clearly. This happens mainly in the aged people after 40. So in aged, yes, people after 40, then they have to wear the spectacles. That's called the presbyopia. Then there is one more, cataract. Cataract is a condition, opaque layer formed on the lens. So, opaque layer formed on lens. As a result, person unable to see the objects. Okay? So, yes, he lose the vision. Person unable to see. So, by surgical method, it has to be removed. That opaque layer is formed on the lens. Number 7, yes, we need to know that, yes, color blindness. Color blindness, mainly, you know very well, it is the X-linked disorder. But it can be, yes, mainly of different type. One is called as a daltonism. Daltonism means a total color blindness. Total color blindness. Person cannot see any color. His life is completely black and white. What he can do? Okay. So it is a kind of a genetic defect. Total color blindness, daltonism. There is one more called as a protomopia. Protomopia is red color blindness. He can see everything but not the red. Red color blindness. Yes, next is a deuteranopia. Deuteranopia. Yes, it is a green color blindness. Then tritanopia. Tritanopia, yes, it is mainly blue color blindness. So these are what you can say various disorders related with the human eye. Okay. So I hope you got any clarity about different kind of disorders. Glaucoma is a condition where excess aqueous fluid that leaks and then enters into vitreous chamber, creating increasing the ocular pressure. Then astigmatism, yes, there is a cornea and lens are disoriented, not proper, so person will not have clarity of the image formation. Then presbyopia, lens will lose its elasticity and then it cannot contract, relax also, person need to use the spectacles, mainly the aged people. Then cataract is a condition where formation of a proteinaceous layer on the lens become opaque or it loses its transparency, there is no chance of image formation, he may become blind. So, color blindness is of four types: daltonism, total color blindness; protanopia is a red color blindness; deuteranopia is a green color blindness; tritanopia is a blue color blindness. So, this is all about today's class. In the next class, we will discuss in detail about the ear. So, students, study well. Take care. Thank you.